Hey bio students, this is Mr. Wolf, and today we're going to talk about setting up monohybrid Punnett square problems. Now before setting up these problems, I'm going to turn you over to my friends at TED-Ed who put together a nice video on some background information.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase y, and the recessive green allele as a lowercase y. The uppercase y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
Awesome video, guys. Thanks for doing that for me. Now in that video you saw that Gregor Mendel was a scientist, or actually a monk, who became a scientist, who actually introduced us to the idea of Punnett square problems. Now, he dealt with a lot of different plant traits. So in this example problem, we're going to be dealing with one of the traits Mendel followed, which was flower color. So make sure before doing these problems you have the handout I gave you in class to follow along with. So setting up a genetic square problem, we're going to deal with purple flowers crossed with white flowers. Now the first thing you have to do in setting up your problem is designate a letter to represent the trait that's in question. So for this example, we're going to use the letter P for purple flowers, since purple flowers are dominant. And so we'll use capital P for purple, lowercase p for white. The next step is to determine the phenotypes of the parents in the problem. So in this example, we have a purple pea plant crossed with a white pea plant. The third step is to determine the genotypes of the parents being crossed. So in this example, we have to learn some vocabulary. In that video, you learned that heterozygous meant that plant had one of each type of gene, a purple allele and a white allele for that trait. Sometimes we use the word hybrid to describe these types of organisms that have one of each type. The other word you learned in that video was homozygous. Now homozygous means they have the same alleles for that one trait. For example, little p would be the homozygous genotype for a white pea plant, and big P would be a homozygous genotype for a purple flowered plant. Sometimes we use the word pure to describe these types of organisms. So in this problem, our genotypes would be big P crossed with a little p. Step 4, we need to determine the possible gametes produced by parents. Now, this is a very important step. This step will actually save you a lot of time when we get into more complex genetics problems. So to do this, we have to understand meiosis first, which we've already watched a video on. So in meiosis, we remember that the alleles for those traits are separated in the production of gametes. So our first heterozygous pea plant would either give a big P to their offspring or a little p, whereas the homozygous white pea plant would only give little p's to all their offspring since they only have one type of allele. This sets up our Punnett square nicely. So the next step would be determining the Punnett square we have to use. For this, we use our gametes. Notice we have 2 by 1. We have 2 possible combinations from parent 1 and only 1 possible combination from parent 2. So in this Punnett square, I need a 2 by 1 Punnett square. So I take the 2 alleles from parent 1, place them in my 2 columns, and I take the 1 allele from parent 2, place them in my 1 row, and now I can figure out the possible gene combinations from both parents. So our kids in this example, or the offspring, will end up being either big P, or little p, little p. In the last step of the genetics problem, we may be asked for a phenotypic or genotypic ratio. What this is asking is, what kind of possible offspring are we going to be having? So the genotypic ratio would focus on what type of genotypes are there. Again, those are the genes that are found in the organism. So we either have a big P, little p gene, we either have a big P, little p genotype, or a little p genotype. It's very important to include the numbers in this. In this case, it's a 1-to-1 ratio of big P, little p to little p. A phenotypic ratio would ask us just what do those plants look like. In this case, it's 1 purple plant to 1 white plant. So in review, monohybrid crosses do follow 2 of Mendel's very important laws. The first law of dominance, which basically says if you have a heterozygous genotype, it would end up taking the phenotype of the dominant trait. So in this example, if you have a big P, little p genotype, those types of plants will be purple and not a mix of the 2. Dominant traits often or always mask recessive traits. And then we have the law of segregation, and this kind of goes back to our discussion on meiosis. What this law says is that the 2 alleles, or the 2 genes for that one trait, are separated during the production of gametes, so offspring only inherit one or the other, never both. So I hope this video helped, and feel free to rewatch this video a couple times to make sure you really understand the material. See you in class.